Hello students, the next topic of our today's discussion is biological oxidation. In this biological oxidation, we have to discuss electron transport chain and its mechanism. So, we have to discuss in this lecture what is biological oxidation and how this biological oxidation affects the electron transport chain and how the formation of this high energy molecules in this reaction here. So, what is biological oxidation? Biological oxidation involves the transfer of negatively charged electrons from one organic compounds to another organic compound or oxygen. So, because here in biological oxidation, we already knows that in carbohydrate metabolism there are some larger molecules are converted into the smaller molecules, and in the conversion of these larger molecules to smaller molecules, there is formation of some electrons here, which is also freely synthesized in these reactions. These electrons are used in these biological oxidation for the formation of carbon dioxide, water and high energy molecules, high energy compounds like ATPs, NADPH, because high amount of energy is also produced during the metabolism of these carbohydrate compounds. So, biochemical reaction, it involves the transfer of negatively charged electrons from any type of organic compounds, which is converted into the another compound or oxygen. In these biological reaction, energy producing reaction is in living cells is directly correlated or coupled with the reduction reaction. So, in any reaction where the oxygen is directly or indirectly lose their hydrogen and or electron here. So, oxygen is directly or indirectly lose hydrogen or electrons and form water and carbon dioxide here, and this biological oxidation reaction involves the involvement of various type of enzymes here, and there are formation of some ATPs molecules in these reactions. And what is the electron transport chain and how they can affect the different high energy molecules, presence of electrons from these metabolic processes. So, what is the electron transport chain? As its name says, it is the chain, it is the pathway which is helpful for the transport of these electrons from different pathways like glycolysis, like citric acid cycle, like gluconeogenesis, glycogen pathways. There are so many pathways which are occurs in our body due to the metabolism of carbohydrate, and the electron which are released in these in the reaction which occurs in the pathways of these important substances, the electron releases from these pathways are transported here by carrier protein, and these carrier protein transported these electrons from these particular pathways and taken it to the mitochondria, and here this mitochondria are helpful for the formation of high energy molecules like ATP, NADPH. So, this is the electron transport chain is the most common pathway in the aerobic cells, where electrons which are synthesized or derived from various substance are transferred to the oxygen here. So, this electron transport chain is a series of highly organized oxidation reduction enzymes, and this electron transport chain is the fourth or largest final stage of aerobic respiration here, because though this electron transport chain, the electron which are needed for the cellular activities are released in the form of ATPs, because all the electrons, all the energy which are released in the metabolic processes during the pathways are carried out in the mitochondria for the formation of ATPs, for the formation of carbon dioxide for the formation of water here. So, this electron transport chain just transport different type of free electrons and convert into them to water molecule, carbon dioxide and ATPs. So, electron transport chain is an oxygen dependent process, because here is this is undergoes under the aerobic condition only. So, these are the oxygen dependent process, and it occurs in the inner mitochondrial membrane. So, the energy rich compounds, what are energy rich compound? Like carbohydrates which having the glucose, fatty acids and amino acids, when which undergoes the series of different metabolic reactions, and they finally oxidize or reduce to the carbon dioxide and water. These reducing equivalents from various metabolic intermediates are just having the NAD positive and FAD, which produce the FADH and FADH2. These are taking the free electrons and form the high energy molecules here. And these two, NADH and NAD and FAD, when produce NADH and FADH2, get reduced coenzymes and pass through the ETC, and then pass through the respiratory chain and electron transport chain, and thus finally it release the carbon dioxide and oxygen to water and carbon dioxide. What is the main passage of electrons? So, when the passage of electron, though this electron transport chain is generally associated with the loss of free energy, because due to the disintegration of larger molecules like carbohydrate, fatty acids, amino acids, proteins, nucleic acid, they are having the high energy, because breakage of the larger bond convert into the smaller bond gives the free energy. Free energy with the loss of free electrons here, which are helpful here, which are transported from that particular pathway to electron transport chain for the formation of this high energy molecules. So, the part of this free electrons which are synthesized in the different pathways is utilized here to generate ATP, adenine triphosphate from ADP and phosphate bonds. So, these are the main reason for electron transport chain here. The location of this electron transport chain is in inner membrane or mitochondria. So, whole mitochondria is responsible for the formation of these high energy bonds. So, the location is mitochondria, and basically they are situated in the inner mitochondrial membrane. So, mitochondria is a center for the metabolic oxidative reaction. So, generally reduced coenzymes like NADH and FADH2, which in turn and utilized in electron transport chain to liberate electron in the form of ATPs. That is why this mitochondria is known as the powerhouse of the cell, because in mitochondria the free electrons, the free energy are utilized here with the help of NAD and FADH2 to form high energy bond, and also carbon dioxide is synthesized here with the water molecule also synthesized. And what is the mitochondrial organization here? Mitochondrial organization having the mitochondria have the 5 distinct part here. The first one is outer membrane, the second one is inner membrane, and the third one is inner membrane space, fourth is cyst, and the fifth is matrix. So, in the structure also determined here, this is the inner membrane, this is the outer membrane, and DNA is also present in the mitochondrial matrix here. Inner membrane space is also present, cysti, ribosome, granules, and ATP synthase particles is also present in the mitochondrial membrane. So, this mitochondrial organization is basically responsible for the formation of this different ATPs or high energy molecules. So, what is inner mitochondrial membrane? Inner mitochondrial membrane here where the electron transport chain and ATP synthesized system are located into the inner mitochondrial membrane. This inner mitochondrial membrane is rich in protein and it is basically impermeable to ions like hydrogen ions, potassium ions, sodium ions, and some nucleotides or smaller molecules like ADP and ATPs also. These inner mitochondria is highly folded to form cristae, and this basically surface area is also very increased due to the presence of this cristae here, and this inner mitochondrial membrane having the specialized particles like lollipops, and they helpful or they are very helpful in the phosphorylation of that subunits, and they are helpful for the formation of ATP. And the mitochondrial matrix which is having the interior ground substance, and this mitochondrial matrix having the full of the enzyme which is responsible for the synthesis of TCA cycle and also helpful for the formation of different pathways, and it is more viscous than the cell cytoplasm and having the less water in their structure, and it provides the very important step in cellular respiration, because it produces it helpful mitochondrial matrix is helpful for the formation of the ATPs with the help of high free energy. So, this mitochondrial matrix is also containing DNA, which is also helpful for the formation of the nucleides here, and it contains the granules of ions which involved in the formation of ionic balance of the mitochondria, because mitochondrial matrix have some types of granules in their structure which is basically helpful for the balancing the normal mitochondrial membrane. And what is the organization of the electron transport chain? Basically, electron transport chain having the 5 different complex in their structures, complex 1, 2, 3 and 4 and 5. There the complex first and fourth are capable of or responsible for carrying the electrons, while fifth is responsible for the ATP generation. And beside these enzyme complex, there are certain mobile electron carriers in the ETC like NADPH, like FADH2. So, these includes the NADH, coenzyme Q, cytochrome C and oxygen, and these complexes first to 4 and mobile carriers are collectively involved in the transportation of electrons, which is responsible for the formation of water from the oxygen. So, most of the oxygen is supplied to the body is utilized in the mitochondria for this electron transport chain. So, what are the component of ETC? There are 5 different components of ETC. Complex 1 having the NADH and coenzyme Q reductase. Complex 2 is the coenzyme succinate coenzyme Q reductase. Complex 3 is the coenzyme Q cytochrome C reductase. Complex 4 is the cytochrome oxidase, and complex 5 is the ATP synthesis. So, these 5 complexes are helpful for the formation of ATPs, formation of different high energy molecules like ATP, NADPH, and controlling the free electrons here. Okay. The component of electron transport chain basically having the nicotinamide nucleotides, flavoproteins, iron sulfur proteins, coenzyme Q and cytochromes. These all are basically responsible for the transfer of electrons from the pathways, and these electrons are ultimately combined with the combined with the oxygen to form water here. So, these components are responsible for the synthesis of water molecule, and these all nicotinamide nucleotides or flavoproteins, iron, sulfur proteins, coenzyme Q and cytochromes all are components of electron transport chain, and they are basically responsible for the synthesis of water molecule here, and they are also helpful for the controlling or releasing the free electrons from the pathways to the electron transport chain. This is the electron structure of the electron transport chain, where these intermembrane space having the transport of hydrogen ions from the mitochondrial matrix, and here is NADH is converted into NADH2, and there is FADH is converted into the FADH2. There is movement of hydrogen ion occurs in the intermembrane space in the electron transport chain, and here the movement of these hydrogen ions from inner mitochondrial matrix to the intermembrane space to the mitochondrial matrix here. So, these hydrogen ion, after transporting from matrix to inner mitochondrial membrane, these are reacted with the water molecules or molecular oxygen here with the hydrogen ion to form a water. So, these electron transport chain having the controlling the different free electrons here, and with reacting these free electrons to the FAD or NAD and form these high energy molecules like FADH2, NADH2 and ATP with the formation of water molecule when converting this molecular oxygen with the hydrogen ion to form water here. Complex 1, NADH dehydrogenase. This complex first, which is already I have discussed previously, this complex first NADH dehydrogenase catalyze oxidation of NADH with reduction of this coenzyme Q here. So, NADH plus hydrogen ion with the coenzyme Q convert NAD positive here, or this coenzyme Q with hydrogen ion here. Here at least 46 proteins with different prosthetic group FMN and iron and sulfur center. So, flavonine mononucleotide is also helpful in the completion of this complex first, and this complex pump 4 protons across the mitochondrial membrane, and this is here in this structure. This is the inner mitochondrial membrane, this is the membrane domain, where in peripheral domain FMN is responsible for the conversion of NADH to NADH positive in the mitochondrial matrix. Coenzyme Q is the benzoquinone nucleotide which is linked to the number of isoprene unit. Here the coenzyme Q or ubiquinone is the hydrophobic in nature, and they are dissolved in the hydrocarbon core of the enzyme. In this coenzyme Q, only the electron carrier, which is not a protein, they are only the protein bound prosthetic group here. So, they have the 3 fully oxidized state here. They have the 3 redox stage in this complex 2 या coenzyme Q here, in which fully oxidized state is ubiquinone Q, और जिसको coenzyme हम Q भी बोल सकते हैं. The second is partially oxidized, which is also known as semiquinone, and which is fully reduced, which is known as ubiquinone. So, they in the complex Q or complex 2 or coenzyme Q, the basically this is a benzoquinone nucleus, and this having the compound which is hydrophobic in nature and which are dissolved in the hydrocarbon core of the membrane, where the electron carrier is not having the protein linked bound prosthetic group. And with the 3 redox state here, the first one is fully oxidized or ubiquinone Q, partially oxidized which is known as semiquinone, and the third one is fully reduced which is known as ubiquinone. Complex second, which is known as succinate dehydrogenase, which is correlated with the Krebs cycle, where the succinate is formed from the formed in the complex second. Here succinate dehydrogenase is the enzyme which used here in the complex second in succinate coenzyme Q reductase in the inner mitochondrial membrane. Here FAD is the initial mitochondrial receptor here, or it is also initial electron receptor, and it is fully reduced to the FAD is reduced to FADH2, where the oxidation of succinate to fumarate in the Krebs cycle. So, the electron released from the Krebs cycle are directly shifted to here for the formation of high energy bonds here. So, the FADH2 is oxidized by the transfer of electron by the coenzyme Q yielding or high energy compounds here. So, this FADH2 is also helpful for the conversion of oxidation of this electrons in coenzyme Q yielding substances, and they cannot pump any type of electron controlling substance or transport of substance across the inner mitochondrial membrane. Okay. And the third is complex 3, which is also known as coenzyme Q cytochrome reductase. It also accepts electrons from coenzyme Q H2 and generally helpful in the electron transfer complexes first and second, and which is also help in the complex third, which is released electron will transfer to an electron sulfur center and then to 2 beta type compounds, which is having the cytochrome first or cytochrome C1. Here finally, the 2 electrons are transferred to the 2 molecules of oxidized form of cytochromes having the free compounds or free protons, which is translocated to the mitochondrial matrix across the total mitochondrial membrane, or this transfer of compounds or this transfer of protons involves the Q cycle, or which is having the proton motive Q cycle, where the conversion of different free electrons from the Krebs cycles, which is transferred to the mitochondrial membrane, and here these protons or these electrons are reacted together here and released with the help of iron sulfur proteins and with the help of these cytochrome C or cytochrome B type. So, this is the complex C or coenzyme Q cytochrome oxidase. The fourth one is the cytochrome oxidase, which is having the complex fourth, which is catalyzes the transfer of electrons from reduced cytochrome C to molecular oxygen and having the 13 subunits. This complex catalyzes the basically the transfer of electrons from reduced form of cytochrome C to molecular oxygen and contain the 13 subunits here, and heme group, which heme group is present in the complex 4 or cytochrome oxidase. Here is the heme A and heme A3. The copper ions which are present in this complex arranged into the 2 copper centers designed as copper A and copper B, and copper contain 2 different copper ions which linked with the bridging disulfide residues, and this carbon A copper A is contain 2 copper ions which linked with the bridging disulfide residue, and copper V is coordinated with the 3 different histidine residues. And in the complex 5, the mitochondrial ATP synthesized consists of 2 mitochondrial subunits, where having the component F0 and F1, which is linked by the Schlerer stack. So, F0 is the electrically driven motor transport which having the lipid bilayer, which channel compound the proton can cross the membrane. So, these F0, F1 and F0 and F1 help in the formation of ATP from the ADP here. So, these are the inhibitors of the ATPs: rotenone complex inhibitor complex 1, amytal carbexin inhibit complex 1, pacrididine compete with the coenzyme Q, antimycin A control complex 3, cyanide, azide and carbon monoxide bind with the complex 4 and inhibits the transfer of electrons to oxygen, and oligomycin is controlling or inhibit the ATP synthase here. So, these are the electron transport chain, its complexes and inhibitors of electron transport chain. Thank you.